Once you know the structure of cholesterol, see question will be to the structure of cholesterol, draw the structure of cholesterol, cholesterol synthesis, all these steps like you have to memorize. 2-acetyl-CoA in the presence of thiolase, acetoacetyl-CoA, from here HMG-CoA, then beta hydroxy, beta methyl glutaryl-CoA, HMG-CoA reductase, mevalonate, 5-phosphomevalonate in the presence of mevalonate kinase, then phosphomevalonate kinase. That means phosphate is being added at each step better. Okay. Mm. No, Faiz. You need not draw these structures at all, but you have to draw the structure of cholesterol. That is the only question which is usually asked. For rest of any of the cycle, you don't draw structure at all. We will go mad learning the structures. Not required. Not at all required. Okay. So this I have confirmed from my biochemistry colleagues. Like students are not supposed to draw the structures at all. Beta, huh? Nothing to worry. Okay. Most welcome. Okay, beta. See, then you have to just do like the names. I will give you one code also to remember this. We reach till mevalonate, right? From mevalonate to 5-phosphomevalonate, only thing you are observing phosphate is being added here. Mevalonate structure, phosphate is added. Hence 5-phosphomevalonate. One more phosphate is added. You see here, you call it as 5-pyrophosphomevalonate. Here one phosphate, here two phosphates. Okay, so again one more phosphate is being added. See, 1, 2, 3. You call it as 3-phospho 5-pyrophosphomevalonate. Okay, then this from here you will see isopentenyl pyrophosphate is formed. Okay, then we have dimethyl allyl pyrophosphate, then geranyl pyrophosphate, farnesyl pyrophosphate. From farnesyl pyrophosphate, we have squalene. From squalene, we have lanosterol. From lanosterol, we have cholesterol. Okay. So these are the steps. I told you I will give you one code. See, first three steps are ketogenesis only, which is quite easy. So there, the code is I don't give funds, still love charity is the code, beta. I don't give funds. I don't give funds, still love, still love charity. Still love charity. You can write this mnemonic better, it becomes easy for you. I don't give funds, still love charity. You can write this mnemonic so that it becomes easy as you just start memorizing. I stands for isopentenyl pyrophosphate. Don't. D stands for dimethyl allyl pyrophosphate. G for geranyl pyrophosphate, funds, farnesyl pyrophosphate. Then still squalene, love, lanosterol, charity, cholesterol. Okay. So you just write the substrate product, enzyme, and where ATP is being added, which mineral is involved. Just convert it into like sentences. Like once you draw the cycle, you can write, for example, 2-acetyl-CoA molecules in the presence of thiolase are converted to acetoacetyl-CoA. That is first point. Second point, acetoacetyl-CoA in the presence of HMG-CoA synthase, it is converted to beta hydroxy beta methyl glutaryl-CoA. Like that, we have certain steps. Okay, you just convert these names into steps. Okay, then I thought I will choose one essay, right? What are the important things synthesized from cholesterol? That also I told you one day. All like hormonal hormones, like steroid hormones are synthesized, no? So what are the steroid hormones we have? Testosterone, estrogen, progesterone, isn't it? Aldosterone, they are all synthesized. This is cholesterol breakdown beta. So synthesis and breakdown, both you learn that would be better. Cholesterol, when it breaks, it forms cholic acid. Just now, when we have studied this bile juice and bile acid, same names came. Glycocholic acid, taurocholic acid, same thing you are finding here. Okay. So these are actually the end products of cholesterol metabolism as well. Okay. So the rate limiting enzyme, that means as long as HMG CoA reductase enzyme is available, you will see the cholesterol synthesis is taking place. So HMG-CoA reductase is generally called as the first enzyme you are finding here. That is the rate limiting enzyme. The availability of this enzyme is essential for cholesterol synthesis. Okay, so that is one essay you can exchange for lipoproteins. Let me think about one more. Tricarboxylic acid cycle. We did in that chapter only. If you choose, glycolysis is over. Citric acid, HMP shunt, glycogenolysis, we are done. Beta oxidation is over. Cholesterol is over. Fatty acid synthase complex over. We will choose one answer from protein metabolism. Beta. Okay. Um, Nucleic acid important topics are also done, like purine breakdown. Okay, let me think which question would be good alternative. Factors affecting enzyme action I did very recently, but still if you want, Rohita wants that answer. If two or three of you wants the same answer, please write yes so that I don't take you to the other answer. I will tell only that answer, please. Shall enjoying you. I will call. Glycine metabolism, I will tell you better.
Hello?
Anisha, we have started class, but I forgot to call you.
Ma'am, I'm busy.
Oh, yeah, yeah, I forgot. Okay, okay, sorry. Okay, bye. Metabolism of amino acids 330. Okay, we'll do glycine metabolism, beta, because that is also an essay question as a substitute. Okay, so glycine, what are the products which are synthesized from glycine? This also I told you very recently. We have production of heme, purines, like C4 carbon, C5 carbon, N7 atoms, glutathione, okay, creatine. All these are synthesized from glycine. So this is one cycle which I want everyone to memorize, metabolism of glycine. So in metabolism of glycine, what is happening? Let us see. What do you mean by metabolism? Here we have two-way cycle. That means we have synthesis and breakdown shown in the same cycle. So let us go slowly and try to understand. See, threonine, it's one of the amino acid. From threonine, you will see the conversion of glycine. Okay, so threonine is the essential amino acid. If you take essential amino acid in the diet, automatically from threonine glycine is synthesized. Okay, this is anabolic reaction, and the enzyme required here is threonine aldolase. Okay, in the presence of this tetrahydrofolate. Okay, so this tetrahydrofolate is actually formed from methylene tetrahydrofolate, N5N10 methylene tetrahydrofolate. For this step, we require serine hydroxy methyl transferase. So glycine can be converted to serine. See, it is reversible reaction. Serine can be also converted to glycine. Suppose if glycine is getting converted to serine, from serine we have the formation of ethanolamine. From ethanolamine we have the formation of glycocolate. From glycocolate we have the formation of glyoxylate. Okay. From glyoxylate we have the formation of oxalate. Then from glyoxylate we have formate. Then we have N5N10 formal tetrahydrofolic acid. Okay. N10 formal tetrahydrofolic acid. This is one way chain we have seen. From glyoxylate with the help of PLP, pyridoxal phosphate. This is active form of pyridoxine. So from this glyoxylate you see glycine is formed. Okay, and this is transamination reaction. That means transfer of amino group from this glyoxylate to the glycine molecule. Okay, so this way glycine is synthesized, hence this is one anabolism reaction. Now we have one more reversible reaction from glycine. It is finally broken down to carbon dioxide plus ammonia. So if ammonia molecule is released from glycine, it will enter into which cycle? Urea cycle. Okay, for which we know tetrahydrofolate is again converted to N5 and 10 methylene. Same reactions, many places we are getting. For one carbon metabolism, these are the things which are being utilized. Okay, NAD can be converted to NADH, NADH also can be converted to NAD, and the enzyme involved here is glycine synthase. That means carbon dioxide plus ammonium, in the presence of glycine synthase, glycine can be produced. Okay. So these are the steps beta glycine metabolism. That's why in this picture, what is written metabolism of glycine. That means it's not written glycine catabolism or glycine synthesis. It's two-way picture which you have to actually practice. Okay. So what are the products which are synthesized from glycine is one part of the answer, and these are the various products which are produced from glycine, like anabolism and catabolism. Both are seen together, we call it as metabolism. Okay, so the important products which are produced is it glutathione here? No, not. So only this much you try to remember, beta. This is some different cycle only, not here. This cycle you can remember. Glycine is also utilized for the formation of creatine. This is important. So here glycine is combining with arginine to form guanidoacetate. From guanidoacetate, you see SAM, one methyl transfer is taking place. S-adenosyl methionine is getting converted to S-adenosyl homocysteine in the presence of guanidoacetate methyl transferase. Okay, so guanidoacetate is converted to creatine, creatine is converted to creatine phosphate in the presence of creatine kinase. So these series of steps you have to write for metabolism of creatine. Creatine can be converted to creatinine. We know creatinine is excreted in urine. Even creatine phosphate also can be converted to creatinine, which is excreted in urine. Okay, beta. So this steps I want you to remember. First, this side number one is this. What are the products which are synthesized from glycine is the question. Then metabolism of glycine is this cycle. Then synthesis of creatinine is this cycle. Okay. So all these things I want you to learn, and then one clinical aspect you can write glycinuria. It's a very rare disorder. So where the glycine content in the urine is increased. Okay. So that is, it is believed that glycinuria is due to defective renal reabsorption. That means glycine is not reabsorbed in the renal tubules, hence it is excreted in urine in large quantities. That condition is called as glycinuria. That is more than enough for this alternative part of essay. Let us see the other questions. Porphyrias we did, competitive inhibition we did, calorific value I will take you, detoxification, functions of oh, many questions are repeated. So from each chapter, porphyrias in that chapter, important questions are covered. See, so many questions are coming as covered within a span of 10 days. So if you really put your sincere efforts, you will definitely pass better. Okay. So instead of porphyrias, I'll take some other question because almost all questions are repeated now. Mm, From digestion absorption, I'll take one question. Okay, jaundice you want me to take? Okay. Yeah, you can suggest some questions like that, beta. Nice, thank you, beta. So one question I am taking from digestion and absorption, beta. From here also, you get many questions like digestion of lipids may be a question. Okay, so when a question like digestion of lipids come, then what you have to see, that means lipids we are taking in the diet, isn't it? So in the form of fat, oil, like butter, whatever we cook, so that is all fat only. So that it is in the form of triacyl glycerols, phospholipids, cholesterol, esters. So they are not changed in the mouth because we do not have enzymes to break these fats. So it is going into the stomach. Even in the stomach, we do not have enzymes to break down these lipids. So hence it is entering into small intestines. So we know in the small intestines we have pancreatic juice, bile juice, and intestinal juice. All these three juices will contain something like pancreatic lipase, phospholipase A2, cholesterol esterase, which will convert triacyl glycerols to monoacyl glycerols. Phospholipids are converted to free fatty acids and glycerol. Okay, and cholesterol is formed from cholesterol esters. So these are the end products of digestion which are observed in the wall of small intestine, and they are transported via portal vein. Enterohepatic circulation. Okay, through enterohepatic circulation, you will see the all these end products of digestion are transported via portal vein to the liver and stored in the liver. Okay, beta. So that is intestinal absorption of fat, digestion of lipids. Okay, now apart from that, now let us do jaundice. So that is one substitute. Rohita, you can please give me one more substitute question also, beta, because many questions are repeated here. Hemoglobin and porphyrias, there we will see jaundice. Nice, very good question, beta, you gave. Degradation of heme, I was actually trying to recollect this picture only. For physiology also, for the degradation of heme, I want you to write this cycle only, beta. Bilirubin, biliverdin, then glucuronic acid, and all this cycle is applicable both in physiology and also in biochemistry. Okay, then. Now, what is jaundice? So, suppose if these bilirubin and biliverdin pigments are excessively produced in the blood. Okay, that means the normal lifespan of RBC is 120 days, but suppose if more number of RBCs are being broken down at a short span, like you see in sickle cell anemia, or maybe there is some underlying pathology because of which the turnover of RBC, that means the breakdown of RBC has increased. So obviously the bile pigment levels also will increase in the blood. Bilirubin, biliverdin levels will increase. That will lead to a condition called as jaundice. Normal serum bilirubin level should be 0.2 to 1 milligrams per deciliter. When it is beyond that, generally you call it as jaundice. And this condition can be also called as hyperbilirubinemia. Okay, so hyperbilirubinemia. Hyper is more, bilirubin, emia is blood. Okay, so basically we have three different types of jaundice. I'll tell you, I think we have a table for that. Okay. If not, also I'll tell you. So the three basic different types of jaundice we have are pre-hepatic, hepatic, and post-hepatic jaundice. Pre-hepatic jaundice can be also called as hemolytic jaundice. Okay, hemolytic in the sense here the lifespan of RBC has been reduced and more number of RBCs are being broken down. Best example is sickle cell anemia, incompatible blood transfusion, malaria. So in all these conditions, you will see RBCs are being broken down more. So that type of jaundice is called as hemolytic jaundice. Okay. So obviously in this case, you will see serum bilirubin levels are increased, but this bilirubin is not combining with UDP. If it is not combining with the conjugating agent, what we have studied in xenobiotics chapter, if UDP's combination is not there with bilirubin, you call it as unconjugated bilirubin. So in case of hemolytic jaundice, we find unconjugated bilirubin is increased in the blood, also in the urine. Okay, and the fecal matter will be dark brown in color due to high bilirubin levels. You call it as stercobilinogen. Basically, the bilirubin is being conjugated in the liver with the help of UDP. You call it as bilirubin diglucuronide. This is the conjugated bilirubin. But in case of hemolytic anemia, the bilirubin remains in blood only. It doesn't get a chance to enter into the liver. Hence, it is not conjugated. We find unconjugated bilirubin levels are high in case of hemolytic jaundice. Second type of jaundice we have is hepatic jaundice. Hepatic jaundice in the sense the breakdown of RBC is normal, but there is some problem with liver. Liver is not in the position to handle the like broken bilirubin levels. So in such case, what happens, for example, due to alcoholism, there may be hepatitis, cirrhosis, there can be liver cancer, like a fatty change of liver. In all these conditions where liver function is impaired, you will see both the types of bilirubins may be there. Part of the bilirubin may be unconjugated, a part of the bilirubin may be conjugated. So both conjugated and unconjugated bilirubin levels will be raised in case of hepatic jaundice. Okay, so hepatic jaundice, hepatocellular jaundice is a type of jaundice which is caused due to dysfunction of liver. I gave you many reasons also. Okay, so the next thing we have is obstructive jaundice. Obstructive jaundice in the sense, see here from the liver we have like gallbladder. So that means the bile juice is supposed to be collected in the gallbladder. From there it is supposed to be released into the duodenum via bile duct. I have shown you just in my previous class the diagram, isn't it? So suppose if the bile juice is not being released in the duodenum, there are some gall stones, bile stones. There are some stones, or there may be some cancer in the extrahepatic biliary system. That means in the gallbladder, in the common bile duct, or in the cystic duct, if there is any stone impaction or if there is any cancer, you will see the bile juice will not be able to enter into the duodenum. So in such case also you will see bilirubin levels will rise in blood. Okay, so that type of jaundice is called as obstructive jaundice. Okay, so in case of obstructive jaundice, generally you will see conjugated bilirubin levels will be rising up, and the other enzymes which will help us to diagnose this disease is alkaline phosphatase. And in case of hepatocellular jaundice, we also find SGOT, SGPT levels are increased in blood. So these are the three types of jaundice, beta. Okay, any questions we got? Okay, porphyrias, we have replaced one question. Nutrition, second question we have replaced with jaundice, calorific value. Okay, I'll replace one more question also, beta. Here only I will explain you Vandenberg's reaction. You will understand easily. We will replace one more question. So in order to check this jaundice, we will do one test that is called as Vandenberg's reaction. We generally learn this Vandenberg's reaction in like various tests we have. I think it is given here only. If not, also I will tell you. Organ function test is the chapter where we get this answer. Let me take you to organ function test. It is related to jaundice only. So in case of organ function test chapter, generally we will check the function of liver. We will assess the levels of, just now we saw bilirubin levels are increased, biliverdin levels are increased, SGOT, SGPT, alanine transaminase, aspartate transaminase, alkaline phosphatase, such enzymes are increased, so with the help of which we can assess whether the liver is functioning normally or not. You call it as liver function test. So under liver function test, now I will explain you Vandenberg's reaction. Now listen carefully. So Vandenberg's reaction, basically we have three types of jaundice. One is hemolytic jaundice. Okay, then second we have obstructive jaundice, and third here we are given hepatic jaundice. So in case of hemolytic jaundice, suppose if the patient has come to us with hemolytic, we don't know what type of jaundice it is. You have collected the blood sample, you have separated the serum, and into the serum now we have added Vandenberg's reagent. Okay, so which is nothing but a mixture of sulfanilic acid and sodium nitrate. So now what we are doing is, once you add Vandenberg's reagent to the serum, generally we get violet color. In case of hemolytic jaundice, we do not get immediate violet color. We need to add alcohol. Then if you add Vandenberg's reagent, then only you will get purple color or violet color. That is what is called as indirect positive. Indirect positive in the sense, if you add serum and if you add Vandenberg's reagent, no purple color. But if you add alcohol, then you will get purple color. That means we have unconjugated bilirubin, which is not reacting with Vandenberg's reagent. But if you add alcohol, then unconjugated bilirubin will now dissolve in the Vandenberg's reagent to give the purple color. Okay, that is indirect positive. Only when you add alcohol, it is becoming purple. Till that time, it is not becoming purple. That is indirect positive. In second step, you see, we have direct positive, obstructive jaundice. I told you it is all conjugated bilirubin only. That means you take patient serum and you put Vandenberg's reagent, full purple color you will see. So this is direct positive. In hepatic jaundice, I told you we have both conjugated and unconjugated bilirubin. That's the reason you take in the test tube patient serum, add Vandenberg's reagent, it becomes light purple. But if you add alcohol, then you will see the solution becomes dark purple. That shows it has both conjugated and unconjugated bilirubin. So the same thing is explained in this paragraph for Vandenberg's reaction. Okay, we are checking the presence of conjugated and unconjugated bilirubin, and we are trying to assess what type of jaundice the patient is suffering with. For example, hemolytic jaundice, test tube, serum, Vandenberg's reagent, no purple. Add alcohol, yes, it is purple. You call it as indirect positive. Obstructive jaundice, test tube, serum, Vandenberg's reagent, full purple. Yes, direct positive. It has conjugated bilirubin. Hepatic jaundice, take test tube, serum, reagent, light purple color. Then you are suspecting maybe it is like hepatic jaundice. Then you put alcohol, yes, intense purple. Then it is hepatic jaundice and biphasic reaction. Very simple, read once you will understand. Okay. Detoxification by conjugation also we did better. Functions of vitamin C. What is the deficiency disorder caused by vitamin C? Scurvy, bleeding gums, because vitamin C is concerned with the formation of collagen. What are essential fatty acids? Linoleic acid, linolenic acid, arachidonic acid. Sickle cell anemia also I have explained you in very much clear way. Like instead of normal hemoglobin, you see the shape of hemoglobin is altered because of what is that mutation, no? Instead of glutamate, valine is replaced. Fiber also we did. Allosteric enzymes we didn't do. Epimers also we didn't do. We will do this. Epimers we will see in the first chapter itself. Think about allosteric enzymes meanwhile. epimers will differ the configuration of H plus OH minus ions at one carbon atom. I'll show you and explain, then it becomes easy. Page is not moving, it's taking time. Yeah, beta. See, epimer. What is epimer? If you observe the structure of D-galactose and if you observe the structure of D-glucose, both are similar, mirror images, but they are differing in the configuration around fourth carbon atom. When you check fourth carbon atom, OH is on left side, hydrogen is on right side. Whereas for D-glucose, H is on left side, OH is on right side. So they are epimers for fourth carbon atom, D-glucose and D-galactose. When you compare D-glucose and D-mannose, they are epimers at second carbon atom, because hydrogen is on left side, OH is on right side for D-glucose, whereas it is opposite for D-mannose. So that structural configuration is called epimers. Okay, that is the meaning of epimers. Okay, so what is the next question, beta? Allosteric inhibition. Allosteric enzymes. Okay, 85. Allosteric enzymes. Yes. So enzymes that are regulated by allosteric mechanisms. So for which you should have some examples to understand. So tasty form. See, basically, if you observe this structure, if you understand this, you will understand the allosteric mechanism. So here in allosteric enzyme, you are shown one enzyme which is in taut form. Taut means this enzyme is not ready to catalyze any reaction. Okay. So whereas the shape of this enzyme is in relaxed form, that means this is ready to receive a substrate and to bring about a product. So here, effect of allosteric inhibitor in the sense, if it comes and binds to the enzyme, it will further increase the tautness of enzyme. It will not allow the enzyme to act on substrate. Whereas allosteric activator in the sense, so this is the relaxed form of enzyme and the allosteric activator comes and binds in such a way that it promotes the enzyme functioning. So allosteric activator comes and binds, then the substrate comes and binds here, and you will see